Okay, recording has been started. Okay. Good. So proteins are made of what? What are the proteins made of?
Aminokislоти.
Yes, of course, they're made of amino acid. So the proteins made of amino acid. The enzymes are special proteins. They're specialized proteins that speed up the chemical reactions in the body without being used up. This is the definition of the enzymes, okay? So the enzymes, they are special proteins that speed up the chemical reaction in the body without being used up. So they speed up the chemical reactions in your body, but no usage. This is very important. This is very high yield information. Okay. It's like a tool. They like a tool. The enzymes like a tool because they help to get that job to be done faster. If, for example, you're going to clean your room, and for this cleaning, for example, the um, what was pылесос in English?
Vacuum cleaner.
Yes, for this, the vacuum cleaner is going to help you. So it is speeding up your reaction. It's speeding up your job. Enzymes are like this in our body. Okay. Do you know any enzymes in your body?
Enzymes.
Do you know any enzymes in your body? I know one enzyme, which is inside our saliva. For example, you drink coffee or you eat bread, you eat food. What's going to happen? The saliva is going to release, right? The saliva. You know what saliva? Sluna, sluna. Okay, and in this saliva you have the amylase. Everything which ends with ase, you have the amylase enzyme. Everything that ends with the ase is called the enzyme. Okay, so you release the amylase from the saliva. Its function is to break down the starch, крахмал or углевод, into the monosaccharides. It can be, for example, into sugar quickly. So you break down the carbohydrates into its monomers. So you're breaking them down. It is speeding up the reaction. If you don't have any enzyme in your body, everything that you eat will be digested maybe a month, maybe one whole week. Okay. So because of the enzymes in our digestive tract, we always speed up the chemical reactions. We have thousands of the biochemical reactions in our body. Okay, so uh, enzymes have the 3D shape. As you can see here, this is the 3D shape of the enzyme. So the proteins are going to, this is the 3D shape of the enzyme. So these proteins, because proteins fold into complete three-dimensional shapes, this 3D shape is very crucial because it determines how the enzyme works. This look, for example, this kind of look only does one job, specialized job. It doesn't do another kind of work, okay? This kind of structure, this kind of structuring of the protein in the 3D shape determines how the enzyme is going to work. Is it going to break down the carbohydrates or proteins or lipids? It doesn't matter. Or it releases acetylcholine in the neurons, right? So every 3D shape, every shape of the protein is important because it does specific job according to the shape. Okay, good. Any question? No question?
No.
And secondly, the heat, the nature of the three dimension of the protein. What the nature means? What the nature means? Who knows? The nature means денатурация. Вы читали об этом когда-то? Что такое денатурация?
It's a changing of the natural structure, yes?
That's good. You're changing, you're breaking down the protein. This is called the denaturation. For example, the egg and the boiled egg. What's the difference between the egg, fresh egg, and boiled egg? The function is in the boiled egg,
Boiled,
you heated the protein, you heat it. After the heating, it loses its 3D shape. It loses this kind of shape. The protein is going to lose its shape. So if it loses its shape, it loses its function. So the protein is losing its function, right, in the inside of the egg. So this is called the denature. So heat is going to denature the three-dimensional shape of the protein. Okay, it's going to break down. It's very important. Too much heat, for example, the fever or high blood pressure in your body. When you have the fever, when you have the high blood pressure in your body, can make the enzymes which are inside of your body, which is which is which is made of the proteins, they can lose its shape. So it stops working. If there is no shape, no function. Okay. For example, in a very high fevers, enzymes in the brain, enzymes inside your brain can stop working properly, and that can lead to the brain damage. They can be the seizures. Have you ever heard about the seizures?
mm,
Seizures, come on. Припадки, seizures. Have you seen?
mm,
Um, seizures, приступы, seizures, припадки. uh, Epilepsy. Have you ever heard about epilepsy? Epilepsy, epilepsy
знакомиться.
припадки. When the body, when they, when they, when they lie down, the patient lies down, and total body reaction happens. They're going to contract. The muscles are going to contract, and that person cannot move. The the tongue also goes behind. Have you ever heard about this? This is because of the high blood
Mm,
pressure. The seizures, the epilepsy is going to happen because in a very high blood pressure, we are damaging the proteins. If we damage our proteins, the enzymes are not going to function. If the enzymes are not functioning properly, the brain can also be damaged. Okay. So coenzymes, what are the coenzymes? So coenzymes are helper molecules. Here the coenzyme. So coenzymes are helper molecules that make enzymes work faster or more efficiently. We talked that enzymes are speeding up the reactions. They are catalyzers, right? But the coenzymes make the enzymes work faster. Okay, some enzymes cannot work at all without their coenzymes. This is very important. For example, we have the enzyme which is called pyruvate dehydrogenase, which take part in the cellular respiration, needs coenzyme A to function. Without it, you cannot make enough energy. Okay, it's for making the energy. Without the coenzymes are special molecules, helper molecules, which speed up the enzymes. Without the coenzymes, you cannot make the enzymes work. For example, pyruvate dehydrogenase. We have special enzyme which is called pyruvate dehydrogenase. This is enzyme. It's ending with A. It is important to make the ATP in your body, to make the energy in your body during the cellular respiration. Without its coenzyme, which is called, for example, coenzyme A, the uh, the the enzyme is not going to work. Okay, so they should be the enzyme. So vitamins. What are the vitamins? Sometimes, so vitamins often become coenzymes in our body. If you don't get enough vitamins, some enzymes won't work in your body. You get the vitamin deficiency diseases. Okay, for example, vitamin B1, which is called the thiamine as well, becomes a
Amazing.
coenzyme. This is the vitamin B1, this is thiamine, becomes a coenzyme for the enzymes that break down the glucose. Okay. Okay, so it breaks down the glucose molecule. Okay, vitamin B1. Without its deficiency, without the thiamine, it's the coenzyme for some enzymes. Without it, without the vitamin B1, you cannot break down the glucose. The deficiency of the thiamine is called Wernicke-Korsakoff disease, for example, or beriberi. Have you ever heard about the beriberi?
Yes.
What is it?
It,
This is a thiamine deficiency, right? Right. This is a thiamine deficiency. So what happens? It's
is
characterized by impairment of the nerves and the heart. General symptoms, for example, loss of the appetite, overall digestive irregularities, feeling numbness, weakness in the limbs, right? Do you remember the beriberi? So you stop to break down the glucose. This is the sign of the beriberi. And we have the disease called Wernicke-Korsakoff. Have you ever heard about this?
mm,
This is the neurological disease. So this is called encephalopathy as well. Have three main symptoms. For example, confusion, inability to coordinate voluntary movements, and visual changes. That will you will learn during the medical school. Okay, for now. So know that the vitamins are very essential. And the vitamin B6, we have the vitamin B6, which is called pyridoxine. Have you ever heard it? This is the and use it in the enzymes. So it stimulates the enzymes that make the neurotransmitters. Neurotransmitters the are, for example, acetylcholine. Have you heard the acetylcholine? The neurotransmitter acetylcholine. Have you ever heard?
mm,
So acetylcholine, this is the special neurotransmitter that transfers the input from one neuron to another neuron. This is the signal transmitter. Okay. So without the B6, you cannot transmit any input. So that's why the vitamin B type, B complexes are very essential for your nervous system, for your cognitive ability. Okay. So that's why you shouldn't skip your vitamin. Good. Your body is not the storage. Your body is not the storage. So you don't have the storage for a long time. You're not given the whole life vitamins. You have to take them always because you're a machine. Your brain is working, your heart is working, your neurons working, your nervous system is working. So that's why you have to give the vitamins. They are very essential. Okay. Any other questions?
no.
Any questions regarding this one? So to summarize, the enzymes are proteins in this case that help the chemical reactions in our body to happen faster. So they have the special 3D shapes, right? That let them to grab their target. If they lose their shape, this shape is 3D shape is really important for lock and key model. I'm going to talk a little bit later. If they lose their shape from the heat, for example, they can't work. And the coenzymes are little helpers that make the enzymes work better. And many of these helpers are vitamins. Okay, if you're missing the vitamins, some enzymes stop working, and you can get sick overall. This is the importance of this slide. Okay, any question?
No.
I hope you got my point. And so here, let let me explain what's going on here. So, in fact, that the enzymes interact with substrate. Do you know what substrate?
Mm.
So enzymes actually interact with substrate, the molecule that they act on. So enzymes act on to special molecule. The enzymes and the substrate should fit together like a lock and the key. Okay? The enzyme always is the lock and the substrate is always is the key. Okay? Only the correct key can fit into the lock and can open it, makes the reaction happen. Okay? So the key is the substrate, the lock is an enzyme, and if the lock and key matches together, what's going to happen? The reaction can occur in our body. Okay? Because of their specific 3D shapes, each enzyme can only work with the specific substrate. Okay, because of the 3D shape, we talked about the 3D shape here. Because of their 3D shapes, each enzyme can only work with specific substrate, just like one specific key that opens the specific lock. Okay, example, for example, we have the enzyme which is called the sucrase enzyme. Let's say this is the sucrase. This is the sucrase. It's ending with ase. That means that they are enzymes, right? So sucrase enzyme only fits and breaks down the sucrase, table sugar, not lactose or maltose, okay? This is the active site where the substrate is going to bind. Substrate can be examples to the substrates. I might have three examples. For example, my first example is lactose. This is the carbohydrate. And I might have the maltose, and I might have the sucrose, table sugar. The sugar that's in your table, the white sugar, is called the sucrose, right? So what I'm going to do, I'm going to break down the sucrose molecule. I'm going to break down this sucrose. Which molecule can which molecule can stick to this active site? Lactose, maltose, or sucrose? Which one? Which one can sit? Which one can match? These are the substrates. Okay, those are the substrates. What I'm going to do, I'm going to match it. These substances, these substrates should match the active site. Which one is correct match? What do you think?
Sucrose?
Of
Sucrose,
course, sucrose. Of course, sucrose. Because the 3D shape of the sucrose is equal with the 3D shape of sucrase enzyme. Okay, this is the enzyme which is called the sucrase. So sucrose can match ideally here because the 3D shape matched, okay? So that's why the sucrose will be the substrate, and in this case, the enzyme will be the sucrose, okay? Cool? Any questions regarding this? And lock and key model, this is called the lock and key model, okay? Theory, lock and key theory. Any question?
Mm.
I hope you perfectly got this point. So enzymes are known as organic catalysts. What do you understand with catalysts? After 5 minutes, my internet can be turned off. Okay, I will quickly turn it on. The Wi-Fi can switch off. Okay, don't worry in that case.
Mhm.
So, well, what's catalysts? What do you think?
They help reactions
Which
happen faster.
factors? Yes.
They are something that speeds up the chemical reaction, right? So, what's organic catalyst? Organic means something that's made of carbon. You know what's carbon, that ukleerod. Organic catalysts are the catalysts which made of carbon containing molecules like protein. Okay, so organic catalysts are proteins actually. Okay, organic catalyst means protein. So enzymes speed up chemical reactions in our body without changing themselves. They're not going to change. Okay, they're stable. They speed up the reaction. Enzymes speed up the reaction, but they're not going to change its shape. They're not going to change themselves. They're going to. They're not going to be consumed during this chemical reaction. They're not used up. Okay, they just speed up the chemical reaction and no change, no using them up. Okay, without the enzymes, many life reactions would be too slow to keep you alive. For example, we have the special enzyme which is called carbonic anhydrase enzyme. Makes carbon dioxide transport in the blood fast enough to maintain breathing and the blood pressure. So carbonic anhydrase is a special enzyme in the hemoglobin molecule, which transport the carbon dioxide very fast. It's going to remove the carbon dioxide from your body very fast. Without the action of the enzyme, we would die. Why? Because the reactions are going on too slow. We are not used up to this. Okay? That's why they're very crucial. So enzymes are not used up. This is the high field. After helping the reaction, the enzymes remains unchanged and is ready to work again. It's going to act to work again. They are reusable. You can reuse it again and again. Only enzyme can catalyze thousands of the chemical reactions. This saves why? This saves the energy and the resources for the body. If you should make the enzymes again and again and again, you use a lot of energy for making that enzyme again. You use a lot of resources to make that again. You don't have to keep making new enzymes all the time after they use it up. That's why the advantage of those enzymes that they are not used up during the reaction. Okay. For example, the one molecule of the catalase can break down millions of the hydrogen peroxide molecules into the water and oxygen per second. So what's hydrogen peroxide? In our bodies, because of the chemical reaction released, can you hear me? The reactions in a lot of hydrogen peroxide, they are oxygen species, reactive oxygen species, which harm our body. What our body does, we have a special enzyme which is called the catalase. So this catalase is going to break down that hydrogen peroxide. It's going to break down that hydrogen peroxide into the water plus oxygen. Okay, so this is harmful for our body. They are useful for our body. So enzyme here is catalase. So it is helping us. Without it, one molecule of the catalase can speed up, can reuse the hydrogen peroxide, can break down the hydrogen peroxide thousands of the times. We don't have to make thousand catalases. We just need one, and it's going to do it again and again. Okay. So what's here? The active site is what is the substrate? If the substrate is going to bind, the substrate is going to bind to the active site of the enzyme. Okay, main working area of the enzymes is active site. Okay, can you hear me?
Yes.
Main working areas of the enzymes are active sites. Okay, so in the enzyme, which is called the DNA polymerase, we're going to learn all those enzymes during our courses. It's okay if you don't know it now. For example, DNA polymerase, the active site, holds the DNA strand tightly while adding new nucleotides during the DNA replication. Okay, the active site is going to hold. And we have the allostatic site. Allostatic site, what's the meaning of the allostatic site? So allostatic site is actually the different spot on the enzyme. Regulators can bind here to change the enzyme shape. We have some regulators in our body. If they sit to the allostatic site, this active site is going to change and another substrate is going to bind here. Or it can either turn on that enzyme or turn off this enzyme. Okay? In the feedback inhibition, for example, the end product of the reaction binds to the allostatic site of the first enzyme, stopping the whole pathway. For example, ATP inhibits the enzyme when the energy is already enough. Do you understand me? So, for
Yes.
example, it's making this enzyme is making the ATP enzyme. It's making the energy, let's say energy. So if you have full energy, if you have a lot of ATP, this ATP is going to come here and sit, and this enzyme is going to stop. It's going to stop making energy. This is so beautiful process. This is how beautiful we are structured. Okay, this is just the magic. Any question?
No.
Uh, for the summary, enzymes are special proteins. They are special proteins that help the reaction in the body to happen super fast. They grab their matching molecule, right? They're going to grab their matching molecule substrate, which we call the substrates, just like a key fits into the lock. That's why it's called um, lock and key theory. Okay, the working area is always called the active site. This is important. Enzymes don't get destroyed during the chemical reactions. Okay, they can keep working again and again. Some spots on the enzymes, for example, which is called the allostatic site, are used to control if the enzyme should work more or should the enzyme should stop. Okay, got this point?
Yes.
Okay, you're so smart. Good. If you don't have the questions, I have a lot of questions to you for today. Okay, so how many students we have now? Jahangir, Muniz,